phylogenomics. This is a little bit specialized topic as well. It's not every people will use it, but I think it's a new branch of the phylogenetic analysis, and it deserves some of the attention to it. And some of the principles can be applied to many others, even though you are not working on genomic scale things. To study evolution on a genomic scale has a lot of applications and also a lot of different approaches. And phylogenomics is just one of them, to use the genomic data to figure out the phylogenetic relationship and or associated topics. So we are not going to talk about some genomic scale study of codon usage or some others. This has been covered, I think, by Dr. Ding as well, and not a focus here. So we are still focused on the phylogenetic reconstruction, and there are several reviews you can look at. Although there are some new methods and new approaches, and but basically the foundation are these three papers in the middle during 2005, and there's annual review papers, and which is quite nice if you are interested in that. Currently, when we want to do the phylogenomics, we have several different approaches. Mostly used one are the first three, and there are still some people use the ones at the bottom. I will very briefly introduce this part a little bit. But we're still working on the primary sequences itself, like the nucleotide or the amino acid sequences. So some people, they will create a super matrix to combine the matrix, or just to combine the trees itself and bypass the matrix. So it's called the super matrix methods and super tree methods. And there's increasing use of the NGS data for to put into the phylogenomics, and we'll talk about that a little. And some people, they use the whole genome features, but not the primary sequence data, like what are the genes, what category of the genes they have, and what is the order of the genes, and or something else, and to have a very broad idea about the relationship among the taxa. There's a different approach.